Hello, this is Tommy. Welcome back to Chatomics. So in today's video, I'm going to share with you how to create pseudo bulk from single cell RNA sequencing data. I will also show you how to use Harmony to correct batch effect. So make sure you stick to the end. So first of all, what is pseudo bulk? So many of you have probably heard of bulk RNA sequencing. And single cell RNA sequencing can profile the gene expression at single cell resolution. And according to this paper, for differential gene expression, pseudobulk seems to perform really well. And to create a pseudobulk, you can simply artificially add up the counts for cells from the same cell type of the same sample. So in this blog post, I will actually show you how to create a pseudobulk from single cell RNA sequencing experiments. So to demonstrate this, I'm going to use the data from this paper, a pan-cancer single cell transcriptional atlas of tumor infiltrating myeloid cells. So it was published in Cell in 2021 and from Zemin Zhang's group. And this is the geo accession number, and we will actually fetch data from FTP using command line. So on command line, you can use wget. So essentially, I will actually have this R Markdown file in the description of this video, so you can follow the tutorial by yourself. So essentially, you just wget and download all the files from this geo accession. And if you look at the FTP, you see there are many files here. The expression.csv, normalized expression.csv, and also the metadata. And it has multiple cancer types. For example, ovarian cancer and pancreatic cancer. So first of all, let's download the data using this wget commands. And I have already did that in my on my local computer. So look at here. So those are the files that I downloaded. And it was actually a zip file, so I already unzip it by using this command, ls gz file, then xargs zip. So it will zip all those, unzip those .gz files in parallel. So it just this is a Unix trick. So the first thing after you download the files, you always want to look at the file content. So on command line, usually you use the last command. So this is just the annotation file, the metadata file. So each row will be one cell and some other columns there. And if you look at the normalized gene expression count, and you see those are the normalized gene expression. Each row is one count and each column is one gene. Each row is one cell and each column is one gene. And you see those are normalized expression because you see those digits there, decimals here. So, but really we want to actually convert those normalized expression data to raw counts. So usually the gene expression data, they are log normalized, and we at Immunitas, Matt Bursting wrote this like small utility to revert the normalized count to the raw counts. So you can actually directly come to this link and download this Python scripts. So essentially the normalized counts will be the counts of that gene in that cell divided by the total number of counts times the scaling factor, usually is 10,000, and plus 1, then log transform it. So, but we really don't know the total number of counts in each cell. So really in this small program, you use this binary bisection action method to find the total number of counts in that cell, and we reverse it, and this assumes the minimum count non-zero counts in each cell is 1. Okay. So you can use this commands, this Python scripts, and you can specify the total number, the scaling factor, and the input, the normalized expression file, and output will be the raw counts. And this is just a trick to use GNU parallel to do this conversion for all the these files. Okay. Of course, you can also do that manually one by one. Okay. And you see here in the command line, you have this normalized expression file that I downloaded. But this is the raw count that is converted by this Python script. Okay, and if you look at the file, the in content, and then you see now those are like integers, like 1, 4, 5, 3. So you know the program probably worked. So press Q to quit. Okay, once we have the raw count, now we can switch to R and load the data into R. Okay. Now I will go back to the RStudio. Okay. So first of all, we need to actually read in the count file and also the metadata file. Okay. So I wrote a small function here. So read the CSV file, turn this as a data frame. And the first column is a cell. If you look at the raw counts here, the first column is the index of the cell. So you just read that count matrix. And the counts file, we can list off them here. So if you do list this, and you see all the counts, raw counts files here. But just for demonstration, I only use the last 3 files because the data is too big and it takes too long to compute. But in reality, you should include all the data here. So that's why I only use the last 3 files here. And of course, then we look at the counts file. So those are the path of those 3 raw counts. And I also give the name, essentially the cancer type name for each file. And now I will use the map function. So counts file is a vector of 3 path, and I use this read counts function that I defined here to read in each of those counts. Okay, and if you look at this counts, it will be a list, and then it will be a named list, and you have 3 these cancer types there. And you can look at the content of the first 5 rows and 5 columns. Okay, that's pretty neat. Now let's read in the metadata file for each cancer type. So similarly, we write a function to read the metadata file for each of the file. So read CSV and put the row names as the cell name, and then return the data frame. Okay. And similarly, we also only use the last 3 metadata files. So if you run this, those are all the metadata files, and we only use actually the last 3 of them. Okay, for demonstration. And again, I also give them a name as the cancer type, and really those lines here is to just use the regular expression to extract those cancer types. So I can use the per function again, map, so the meta files over here, and reading all of them. Okay. So meta will be a list as well, a list of 3. If you look at then you see, okay, this is just the metadata file for PAAD and the first 6 rows here. Okay. So one thing I noticed that in this data set, different count table, they have different number of rows, meaning they have different number of genes. So if you look at this, you map function and counts, this is a list, remember, then you n row the number of rows here for each of the count matrix here. So you see they have different number of genes. So most likely those data sets are from different studies, and different studies may have different number of genes included. Okay. So how many of them are how many genes are common? So you can use this function map and row names and use reduce and intersect. And this should be the per function. So 13,000 genes, they are common for all 3 data sets. Okay. Now we can make a Seurat objects. So you can use the map2 function and you provide 2 arguments, x, y. So x is the counts, a list of 3 and count matrix, and meta, and a list of 3 data frame with the row names of the cell names. Then you can use this create Seurat object, and then .x refers to the count, every element of the counts, and .y refers to every element of the metadata. So object will be a list of 3 Seurat objects, as you can see here. Right? Okay, and in the original data sets, actually they also have like normal, adjacent normal tissues. So, but in this case, we only want the tumors. We can just subset the tissues equals to T, meaning tumor, for all of them. Okay. And you see actually the number of cells will be reduced. For example, here UCAC was like 4,724 cells, and oh, this is actually the same here. How about, oh, sorry, because I actually didn't rerun this on the fly, so this is already the filtered Seurat object. Okay. So anyway, so let's write another function to do pre-processing the Seurat object. This is just standardized process. Normalize data, find most variable genes, scale the data, run PCA, run harmony, and run Heatmap, and find neighbors and find clusters. So this standardized process. And so we can just run this using the map function and loop through all the objects and run this pre-process Seurat. And then we can, just for one of them, and we can plot the dimension plot, so essentially showing the Heatmap. So this is Heatmap, and then those cells are colored by cluster ID, and P2 will be clustered by the annotation from the original author, major clusters. And we see, looks the cells, they are actually colored by the original cell annotations. There's some mast cells and some p-dendritic cells, classic dendritic cells, CDC1, CDC2, and CDC3, and some other monocytes and macrophages. Okay, now let's actually create pseudo bulk for each of them. Okay, so to do this, we first need to actually clean up the annotation a little bit. So let's use this to check. So we can take a look at all the annotations for those 3 cancer data sets. And you see this actually is the same, but sometimes the prefix is a little bit different. For example, you can see M09 macrophage C1QC, and in a different data set it's called M10 macrophage C1QC, and here is M11. So we want to just clean up this annotation a little bit, just remove this and only use this as a new column called annotation. So this is essentially what this function is doing here. And so we can again run the map function objects, and it will run this clean cluster name function for each of the objects. And if we do that again, and you'll see the annotations now you don't have that M0102 prefix, so they are kind of consistent across different data sets. So far you have seen I frequently use this map function from the per R package. So one of the key things that computer is good at is to do repetitive work. So if you find yourself keep like copy pasting the same commands, you want either use the for loop or use the per map function, so to avoid repetitions of your code. So learning how to use per is definitely a life-changing event for me. So make sure you Google around and look for the tutorials. Maybe in one of my future videos I can introduce how you can use per. Okay, now let's create a pseudo bulk. Okay, there are many ways to do it. So let's use the Presto package for low level analysis instead of using the wrapper function in this tool called MMuseCat. It's a bioconductor package, and it has many wrapper functions, but then you will need to convert the Seurat object into a single cell experiment object. Okay, let's use Presto. And first of all, let's look at the metadata on one of the Seurat object. Okay. So we will need to create a pseudo bulk per cancer type, per sample, and per cell type. And those are in the these 3 column names, like annotation here, cell type, the patient, which is a sample ID, and then the cancer type. Okay, so in Presto there's a function called collapse counts, and you give the raw counts and the metadata and the column name of the metadata that you want to create a pseudo bulk on. Okay, let's do this. And these pseudo bulk objects, again, you use the per function map objects and create pseudo bulk. Okay. So let's look at the number of genes again. As we have seen, they have different number of genes. So let's actually intersect only the common genes, so we can actually put all those 3 data sets together into a single data matrix. So what we do here, we use this function per reduce, and this is all the gene names into a list of 3 here. So if you look here, this one, it will contain all the actual genes in the first data matrix. And what you can do, you can use the reduce function and give the list of all the genes in 3 data sets and use intersect. So it will actually iteratively intersect all of them. So you look at common genes and the length. Now you have 13,000 something. Now we can subset the matrix. Okay, so the common genes, the rows, and then we just return the matrix. So this is a really simple function here. And then we can again use the map function, the pseudo bulk objects, and then subset the common genes. Okay, if we do that, look at let's see here. Mets. Okay. And then the same thing, like we actually take out the metadata into the same list of metadata frame here, So this will be a list of 3 as well. So let's just look at one of them. Yep. Then this is the data frame, the count matrix for PAAD. Now let's do a principal component analysis. First of all, we need to actually merge all the count table or count matrix into a big one. Again, you use the reduce function because mats is a list of a matrix. Use the column bind, cbind to actually bind them together, because now the row or the genes are the same. You can just concatenate them together using cbind. Okay. And you use rbind to actually stack all the metadata together. Okay. So you look at the dimension. Okay. So we also need to actually so this is the final mats on the matrix. So you have like 13,000 genes, and columns will be a pseudo bulk for each sample, and for each cancer type, and for each cell type as well. Okay, and here you just do a little bit normalization. So get the column sum of those genes in each pseudo bulk, and then you just divide it by the total number of reads for each pseudo bulk, and do a log2 transformation. Okay. And once you get this, now you can do you can first find the most variable genes, because you don't want to use all of them. Use gene filter function, and you give the matrix and use the row VAS and sort them and get the top 1,000 genes. So if you do this top 1,000 variable genes, now you can subset the matrix using only the top 1,000 genes. And now you can do PCA. So you can use the principal component analysis. So use PRComp. So transpose the subsetted matrix, count matrix, normalize the count matrix, and center them, and also scale it. So if you're new to PCA, you can read my old blog post here. So again, now once we calculate the PCA, we just extract from the PCA objects the first component, PCA component, and the second principal components, and also bind the metadata together. So if you look at data frame, will be like this. So each row will be one pseudo bulk. So the first column will PC1, PC2 coordinates, and it will also bind the metadata, so an annotation, patient, and cancer. Okay. Now we can plot the PCA. So it's also just simple ggplot2 functions, and now we color them by cancer type, but we can also color them by the cell type here. So the total 13 different cancer cell annotation cell types here. So I used this from polychrome to create 13 actually unique colors. But the first color is white, so that's why I use 14 and then remove the first color. Like this. And then, but if you use this, the first one will be white, so I remove this one. Okay, and then use the ggplot2 function, and then now I color each point by annotation. So if you look at them, P1, so you color them by cancer types. They're kind of mixed together, different cancer types. But if you look at P2, the similar same cell types, they actually cluster together. Again, here each dot here is a pseudo bulk sample. Okay. And we can also visualize them in a heat map. Okay, so those are simple heat map functions using a complex heat map. So I really highly recommend you to learn how to use complex heat map, because it's really the best package to make heat maps. So I add a little bit top annotations here, and I can create the heat map here. Because it's actually 1,000 genes here, so 1,000 rows here, so it takes a little bit of time for heat map to draw. But again, you see, maybe we can actually go back to the rendered version here. Okay, we have talked about here, create a pseudo bulk. And this is the heat map. So each row here will be one gene. So each column here will be one actually pseudo bulk sample. And there are two bars here. This is the cell type annotation, and the second, this is the cancer type. And you do see some structures here. For example, mast cells, they tend to cluster together for pseudo bulk. And again, those are from different cancer types or different data sets. They're like batch effects, and in my experience, even within the same cohorts, if you do single cell RNA sequencing, and within the same cohorts, sometimes you also see the cell types, they're actually separated, and the cells are clustered by sample, not by cell type. So in this case, you can use Harmony to correct the batches. So Harmony is a single cell integration tool that's really fast and efficient. So in this case, we can use the Harmony package, and we can give it the count matrix as the input. This is the metadata column, and then you specify the column name that you want to actually correct for batches. So in this case, it will be the cancer type and also the samples. Okay, and so then again, after we correct the principal components using Harmony, and we plot the PCA. Okay, actually not we can plot the Harmony space. So again, you'll see here, this is a Harmony 1, this is Harmony 2. So essentially those are the PCA, PC1, PC2 that is corrected by Harmony. And you see here, now it looks like the similar cell types, same cell type, they even cluster better than the previous one here, like here. So to better visualize it in a heat map, you can actually, oh, by the way, we see this different cell types, they come separate, and then similar cell types, cancer types, they're separate from each other, and the similar cell types they cluster together, which is expected because Harmony artificially mixed the samples from different batches. So, but then just be aware that any single cell batch correction or integration tools will most likely remove some of the biological signals. So there's a recent paper from Nancy Zhang's group. They actually built upon Harmony and created this new tool to recover the biological signals from after integration. Okay, I hope you can read this paper on bioRxiv. So we can also visualize the Harmony corrected samples by calculating the correlation of the Harmony embeddings. So the same Heatmap function here. Now you see this annotation, which means the cell types. This actually looks much cleaner than the previous ones. If you remember, if you look at here, it's a little bit more messy here. So, which again is expected because Harmony trying to actually squish different samples of the same cell types mingled together. Okay, so I think that's it for today. I hope you learn how to construct a pseudo bulk for single cell data, and there's some further learnings like this blog post by Jim Fan. You can create pseudo bulk just by linear algebra, and in bioconduct there's a tool called Scran pseudo bulk DGG to create pseudo bulk and perform differential gene expression analysis. Of course, we can also use MuseCat. Okay, that's it for today. I hope this is useful for you, and make sure you subscribe. See you next time.